Yes, I do feel a little fraudulent presenting after Camille and Alison at a mycology conference. But I do still consider myself a botanist. Like when the census comes around, I put occupation botanist. I don't fill it in when I'm coming back from overseas because it tends to hold you up at the airport. But it's been a while since I've done a lot of hardcore botany. Most of the time now I'm focused on a group of plants which have always really captured my imagination, and that's the orchids. There's something about orchids that other plants just don't have, They're the pop stars of the plant world, and serious botanists, a lot more serious than myself, actually despise orchids because they're so popular. They're so beautiful, and look at the different shapes and sizes and diversity, and any plant that can trick animals into having sex with it in order to get pollinated is pretty cool. And that's what a lot of our orchids actually do. That, like this Caladinia up in the top, tricked the poor unsuspecting wasp. But we're not talking about that today. Instead, going to talk about fungi, because you can imagine my delight when in 2015 I got a job with the Orchid Conservation Program at the Royal Botanic Gardens, Victoria. Wonderful opportunity. And I remember my first day at work imagining this workplace must just be dripping with endangered orchids, just hanging off the walls. And I started in November, and that time in November in the laboratory where we grow a lot of our orchids, there were actually no orchids there. There were boxes and boxes of this stuff. Fungus. And so today, my talk is how invisible fungi save endangered orchids from extinction, or why is the Orchid Conservation Program lab at the Royal Botanic Gardens Victoria full of fungus? I'd like to start with ice cream. I think a lot of things are improved when started with ice cream, like breakfast, lunch, dinner, to name a few. And as an ice cream connoisseur, I know that there is only one true flavour of ice cream, a flavour that allows the ice cream to truly express who it really is. And that, of course, is vanilla. Vanilla, of course, coming from the vanilla bean, right? Wrong. The vanilla bean is not a bean. It's not in the bean or pea family. It's the seed pod of the vanilla orchid, actually a genus of orchids with over 100 species. Several of which have been commercialised. And there's, and as you're eating your vanilla ice cream, you're eating those little black vanilla seeds, That's the sign of a true high-quality vanilla ice cream in my mind. It's just got those little black vanilla seeds. Now, there's two things that strike you about vanilla seeds, and one is how delicious they are, and the second is how tiny they are. Like, they don't look like real seeds. This is a real seed. Any wheat farmers in the room will know that. There's a grain of wheat, right? So a grain of wheat, like most normal seeds, is made up of 3 bits, roughly. It's got like the shell around the outside. It's got the little embryo. That's the bit that actually starts growing. And then it's got this like 95% of the seed, which is called endosperm. It's the carbohydrate-rich food body that feeds the embryo and gets it to germinate. It gives that embryo enough energy just to put up its first little leaf and first little root when it can start photosynthesizing and making energy for itself, and it's away. Orchid seeds, though, they don't have that, right? These are orchid seeds. Now, to give you a scale of context, this circular dish that these orchid seeds are in is about 5 centimetres across, right? So they're tiny. And if we zoom in on them about a bazillion times, you'll see that orchid seeds look very different to a grain of wheat or any other normal seed. They've got an embryo in the middle, and they've got this seed coat more or less around the outside, but they don't have any endosperm. So they can't germinate, right? And all of a sudden it's completely obvious why there's so many endangered orchids in the world. These stupid things. So I thought we'd start with a mindfulness exercise. This is actually not really a mindfulness I don't actually know what mindfulness exercises are, but I imagine they go something like this. Okay, so I'd like everyone to imagine that we're not people, we are in fact orchid seeds. Okay, that's slightly difficult because orchid seeds are about 500 micrometers tall, which is about half a millimeter, and it's, yeah, life is hard when you're only that big. And this, we're sitting in a room. We're not in some footy rooms at a footy ground. This room is actually a seed pod, right? So our flowers flower during the spring, got pollinated, the petals were drooped, the ovary of the flowers swollen, and then as summer sort of threatened, the soil's dried out and the seed pod is split open and poof, out we go into the world. Now, when you're only 500 micrometers tall, you tend not to weigh very much, so if there's a strong northerly wind blowing, there's a chance that some of us may end up in Bass Strait, or worse, Tasmania. But if it's just a gentle breeze, then most of us will probably land about a metre or so from the parent plant. And if we're lucky, maybe in a little scratch in the soil where a bandicoot or a bird or something has just scratched and broken that soil crust and enabled us to get in contact with the soil proper. And then we just sort of sit and bake through the summer. But eventually, the autumn winds will come, or autumn rains will come, and the soil, as we know now, because we've had some actual real mycologists talk to us, comes alive with fungal hyphae, and they wake up from their summer slumber and start rummaging through the soil. Is that the noise that fungi make? Can you? I've watched too many David Attenborough documentaries. They're eating like dead leaves or dead twigs and all sorts of stuff. I'm sure they're delicious. And we're just sort of sitting there and we hear this like, oh no, we've been discovered. The fungi have come for us. It's about this time in an orchid seed's life where you start to reflect on your life's achievements and put the finishing touches to your will, which as something 500 micrometers tall is probably not a very extensive document. But this fungus in particular, at this particular spot, on this particular day, doesn't eat us. It drills through our seed coat, and its fungal hyphae grow into the cells of the embryo, and instead of eating us, start to feed us. They feed the embryo water, nutrients, carbon, energy, and germinate the embryo. The embryo swells, its cells divide. Up one end, they get a bit pointy and start to make a leaf shoot, which actually becomes a leaf, and up the other end, a root that sends its dropper down into the soil. And hopefully, if the season is long enough and reasonably good, can make a little tuber and survive the summer. I think that's just an amazing thing, you know? Like how did it come to be that plants just totally gave up on being able to reproduce by themselves and just completely relied on fungi to do all the work for them? It's a beautiful thing. So now, which of these fungi live with orchids? What fungi are doing this? Like, is it all... No. When I think of fungi, this is what I think of. Actually, that's not true. That's more what I think of. But it turns out that the fungi that live with orchids are not... Well, there's lots of different fungi that associate with orchids around the world. But in Australia, the majority of our orchids associate with fungi that are loosely termed microfungi. So these are fungi that don't produce visible fruiting bodies, like they don't produce mushrooms. They reproduce by microscopic spores in the soil, and you really have no idea that they're there. Except that we can grow them in petri dishes, and that's kind of what they look like. They all sort of look much the same. But yeah. Our spider orchids associate with a genus of fungi called Serendipita. That's probably the nicest thing I can think of to say about this group of fungi, is its name. But all of these orchids associate with this genus. Our donkey orchids and sun orchids associate with fungi called Tulisnella. And our leek orchids and green hoods associate with fungi from the genus Ceratobasidium. So there's some trends, there's some patterns, you know, in which fungi associate with which groups of orchids. Now, some orchids, like many of our spider orchids, these ones on the left here, have very specific fungal associations. So the species of orchid will only associate with a single species of fungus. And in fact, many species of spider orchids from the genus Caledonia all associate with the same, with just one species of fungus. It uses all these different, it doesn't matter which Caledonia species you look at from across the country. Probably about a third of the species in the genus Caledonia all associate with just a single species of Serendipita fungus. At the other end of the spectrum, you've got something like the green leek orchid here from the Warby Ranges, which in a population about half the size of this room associates with 7 different fungi, 7 different species of Ceratobasidium fungi. So that's a much more diverse association. And that's nothing compared to orchids from Europe, where orchid mycorrhizal fungi associations have been really well studied. And it's nothing to find European orchids that associate with 20 or 30 species of fungi from different genera completely. So now, if you're only associating with a single species of fungus, you probably want that fungus to be pretty widespread, all right? Otherwise, it's not a very good life strategy. This is the underground orchid, which is a very interesting orchid. It actually lives underground, doesn't produce any leaves, and produces this flower just underneath the leaf litter, at the very sort of surface of the mineral soil. Yeah, very interesting genus of orchids. Now this is a map showing, this is where this underground orchid is found here in this orange dot, and also a few places in southwest WA. These are different species of underground orchids, but all from the genus Rhizonthala. And this is the distribution of its fungus. So the CSIRO did a really big project where they went around the country and dug up bits of soil and ran them all through a DNA testing machine to see what was in there. You know, fungi, bacteria, all sorts of things. And there's a big database that they provided, and we searched for the fungi that live with this Rhizonthala orchid and found that actually it occurs everywhere there's a red dot is where this fungus was found. So we've got a really rare group of orchids, very restricted, that associate with fungi that are found all the way around all across the country. The brighter yellow areas are places where we have a high degree of certainty that the fungus occurs. So you can see southwest WA and southeast Australia, that's where there's a lot of orchids that associate with fungi. We might expect there to be fungi there. But this fungus also occurs right up in the top of the very far north of the country and through the middle. So some fungi that associate with orchids are really widespread. Others may not be widespread, although it's much harder to prove that a fungus is, or anything really that's hard to survey for, is truly rare. So that's all a great bit of information about fungi. Now, one of the things that I think attracts a lot of people to orchids is because there's a lot of rare endangered species of orchids. Victoria has about 400 species of orchids, and about half of them are threatened in some way with extinction, and some of them are on the absolute brink. So the Botanic Gardens has this wonderful orchid conservation program where we grow endangered orchids and plant them back out into the bush and create new populations to stop species going extinct. It's actually the largest endangered orchid rewilding program anywhere in the world. And we grow things like this beautiful Caledonia pumila, which was last seen in the 1930s, thought extinct for 80 years until someone rediscovered it in 2009. And this is in southwest Victoria. And they've only ever found one plant. Someone collected a little bit of seed. We managed to get a few plants growing in the nursery, and now that we think that one plant might have died in the wild. So it may be extinct in the wild again, and it's only persisting in the shade houses at the Botanic Gardens. So it's really important if we're to conserve these orchids to, to grow them in cultivation and ideally to replant them back out into the bush. But how do we do that with a plant with seeds that can't grow by themselves? I mean, you can't just get a bit of orchid seed and sprinkle it on some potting mix because it doesn't do anything. Instead, we have to recreate this orchid fungus association in the lab. So we go out into the wild and we carefully dig down. This is a leek orchid here. This is it's the top of its, or the bottom of its leaf. Goes down, down, down into the soil, and somewhere down there's a tuber and several roots. So we carefully extract, this is a piece of root here of the leek orchid with the scalpel. Dig that up and bring it back to the lab, wash it and put in some bleach to surface sterilise the outside of that root to kill all the other fungi and bacteria and rubbish that grow on the surface of plant roots. And then we put that sterilised root onto a plate, a petri dish full of agar sort of mixture, and out of that root grow these orchid mycorrhizal fungi. You can see a little piece of root tissue has been put on the agar plate here, and these are the hyphae growing out away from that root. Then we added the orchid seed into that petri dish and put them in a box in the dark for a few months, and hopefully when we open the box we see something like that, which is you can see these ungerminated orchid seeds here. This is the little cube of little block of fungi that we've used from that previous agar plate, and the fungus has grown across the plate and inoculated these orchid seeds and caused them to germinate. And yeah, that's a beautiful thing. You can see it a bit better on this photo here. There's a little cube of fungus that we've got, and you can see the hyphae sort of threads emanating from there. Now, the good thing about growing orchids this way is because they produce so many thousands of seeds, you can really scale this up and grow thousands of the things. And that's what we do. We grow them under, specialised growth room, under lights and certain temperature conditions. When they get a bit big for their petri dishes, we put them into these takeaway food containers with a bit of vermiculite on the top, and that gets them a bit bigger, and then they go into pots in our nursery. And in springtime, when they flower, the nurseries at the Botanic Gardens at Cranbourne are incredible. We have, what was the figure someone was talking about the other day? I think over 20,000 plants across 200 species of orchids currently in the shade houses of the Orchid Conservation Program. And it's the largest collection, we think, of endangered orchids anywhere in the country, and probably one of the largest collections of endangered orchids in a collection anywhere in the world by the number of species and number of plants. So it's a truly spectacular site. And for the very first time ever this year, we're opening the shade houses in September. So if you want to come and see just a spectacular display of orchids that you will just never see anywhere else in the world, including some critically endangered species, I don't think it's yet been advertised, but a couple of Saturdays in September this year, there's going to be open shade houses at the Cranbourne Botanic Gardens. Fair way from Euroa, but worth the trip, I reckon. Now, the point of growing these orchids is not to use them as a nice display for people who like orchids, but actually to plant them back out into the bush. So usually every year, we do reintroductions of threatened orchids into places where we've chosen for these orchids to go. Places where the land is protected, the pollinator of the orchid is present, the habitat matches the habitat of the wild sites of that orchid. And we have a huge amount of help from volunteers from the Australasian Native Orchid Society doing this. They do heaps of great work in the orchid conservation space, and this is just one of them. They're always looking for new members, so get in contact with them if you are interested. And yeah, we create these new populations. Somewhere, I think around about 20% of the reintroductions we've done have formed self-sustaining populations. So the plants that we've planted have had babies, and then the babies themselves have had babies. So, and we're getting a lot better at doing this through a heavy focus on research. And so it's relative to other plant reintroductions, this is one of the more successful plant reintroduction programs in the country. And it's really exciting work. Yes, so an example of this is Calodenia colorata. This is a spider orchid from southwest or far west Victoria. We planted nearly 900 plants between 2013 and 2017, 80% survival of those plants, plus another 600 new recruits. So it's proof that, you know, you can bring back these species which were once on the brink of extinction by understanding things like their pollinators, their minimum population sizes, microsite preferences, genetic diversity, and importantly, fungi. So how does understanding fungi help save our endangered orchids? Well, I've got a couple of examples here. This is a leek orchid, Prasophyllum frenchii. Now, that method that I described for how we grow orchids is, we've been doing that for like 20 years with a lot of the spider orchids and the green hoods and the sun orchids and the donkey orchids, but we've never been able to do that with any measure of success with leek orchids for some reason. And this genus of leek orchids is one of the largest groups of orchids in the country. And there's heaps of endangered species, some of which are critically endangered. And if we can't grow them, then there's not much we can do to help them, really. So why weren't they growing? Well, when we did the normal things that work well for the other groups of orchids, this was all that happened. The seed would swell a little bit, but they wouldn't germinate. So we thought, oh, maybe the fungi that work with leek, that associate with leek orchids, have some they just need some. They do things a bit differently. Maybe they have different nutrient preferences. So this was the standard. It was like industry standard. You probably can't see it because the lectern's in front of it. It's like dilute porridge mixture. That's what we used to grow the orchids on. And it worked well for all these other orchids, but not for leek orchids. But when we added in extra nutrients, we got repeated germination of leek orchids. Interestingly, we used 3 different samples of fungus from the roots of this particular leek orchid. And there's 3 different fungi, all prefer different nutrient media. So you can see this, the darkest green fungus here, fungus C we call it, it grew pretty good on a lot of different media, but best was on this high nutrient MOM media. This middle green one here, the B, it hated the MOM, but when you removed the sucrose, then it caused quite high levels of germination. And this pale green 1A, which you can hardly see, actually did best on these bran media here with wheat bran instead of oats. So you can see that different fungi have different nutrient preferences that translate into different levels of germination for the orchids that they associate with. What was interesting is that all these 3 species of fungi had identical DNA sequences for the region that we were looking at. So there's obviously a lot of diversity, even within a species of fungus, that probably in the wild helps that orchid to do better or when under different environmental conditions. Now you can see the germination percentage is only about 5% at the best of times, but that was a plate of this Prasophyllum frenchii, and that's a 5% germination rate, which is more than enough when you're producing thousands of seeds. This is the green leek orchid from the Warby Ranges I mentioned earlier. So it associates with 7 different species of fungi, represented by these orange bars here. So 2 species of fungi were found in the roots of this green leek orchid all through the season. So in autumn, winter, early, late winter and spring. Very common. There were 5 other species of fungi that we only found very occasionally in the roots of this species. So that's interesting because a lot of our orchids, as I mentioned, have quite specific fungi associations. So if the orchid only associates with one species of fungus, then it's pretty likely that that's the fungus that's going to be involved in the germination of that seed naturally. But with an orchid that associates with lots of different fungi, it does make you wonder, well, which of these fungi are involved in germination and what are the roles of the other fungi? I've got no idea. So to work that out, I buried these little tea bags. So you can get that fancy tea that's in those like plastic mesh tea bags. Well, if you'd cut those bags open and pour out the tea and put fungi in them, you've lost some really good tea bags, and I wouldn't recommend it. But you can then bury those bags around the base of wild orchid plants out in the bush in autumn, dig them up in spring, and find these germinating little seedlings here. And if you look, this is a microscopic photo, but this is a germinating seed of this green leek orchid. And you can see these little cotton wool ball shaped things here in the bottom half of this seed. These are the coils of fungal hyphae. The fungus actually has a rusty red colour, so as it gets older, it gets a darker colour. And these are the fungi that are feeding this germinating seed in order to get it to germinate. And when we looked at the identity of the fungi that we found in these germinating seeds, we found that they all came from this single species of fungus. So only one of the 7 species of fungi was involved in germination for this green leek orchid. Now that's interesting from a theoretical point of view, but also from a practical point of view of growing this species and reintroducing it back out into the wild, because it turned out that actually all of these fungi would germinate the seed of the orchid in the lab, which is a bit surprising, but also potentially misleading, because if we didn't know all of this interesting information about which species of fungus associates with the orchid in the wild, and we just see, oh, well, these fungi germinate the seed of this orchid in the lab, we might be tempted to only use a few species of fungi to grow that orchid in the lab. And if we pick the wrong ones, we might actually be reintroducing orchids that lack a critical species of fungus that is essential for that orchid to complete its life cycle and to germinate in the wild. So having a good understanding of the fungi is actually essential for being able to undertake a successful reintroduction of an endangered orchid. I'm almost at the end, but maybe that leaves a few.
Another 5, possibly.
Yeah. No, I'm pretty much done here. I'll just finish with how to get involved in orchid conservation. I know this is a fungi conference. I'm playing to the wrong crowd. In Victoria, I don't want to give you the impression that we've got it all under control and that this wonderful orchid conservation program has everything like totally sorted, because that is not the case. There are so many species of endangered orchids and relatively few people who work on them, that there's just like heaps of orchids that are out there in the bush, a lot of them near the brink of extinction, and just no one checks on them, and they need some friends. So these are some species of leek orchids, which is the genus of orchids that I've spent most of my time working on. And these are the total worldwide populations. This is an interesting one. I included this because it's quite a local species. This species is from Monia North Nature Conservation Reserve, its biggest population, which is a rare reserve out on the plains and one of the best and largest of those precious reserves on the plains around here. This is another local species from near Benalla. This is Prasophyllum gilgai. It was last seen in 2016 with 3 plants, and me and Sally have been looking for it most years since and haven't found it, even after these last wet years. So that's a species we're very concerned about and could possibly be extinct. This may have been the last photo taken of it. And this is another species from Gippsland, near where I'm from, and another species from Western Victoria. This one on the end here, Prasophyllum fosteri, although there's 8 plants, or there were a couple of years ago, there's a local naturalist who lives around the corner in the nearby town, and he gets out there every year and checks on it and collects seed on it. And so we now have a reasonable collection of seed for this species at the Botanic Gardens, and that is the only difference between this species on the left here and this one. This one's almost extinct, and possibly, there's not much we can do about it, we're not sure. But this one is in a much better situation because there's someone who lives around the corner and has checked on it and collected seed from it. So if you're interested in orchid conservation and you can tell the difference between a Prasophyllum and a mycorrhizal, then it would be great to get in touch with me, because there's so many orchids out there that need someone to go and look for them, someone to survey some area of possible habitat, or someone to collect some seed. You can make a really big contribution, actually, if that's the thing that you're interested in. Also, joining the Australasian Native Orchid Society is a really great thing to do, because they do heaps of wonderful conservation work and help us a lot with our reintroductions. And also, if possible, consider helping us out at the Orchid Conservation Program, because we run entirely off grants and donations. We don't actually have any ongoing funding. So yes, that was all I had. Thank you very much.
Marc, wow. I think you played to the right crowd. I'm sure there's lots of questions about orchids, even though it's a mycological argument.
I wonder if you could, I've got 2 questions. One is, could you explain the benefits to the fungus from their relationship with the orchid? And secondly, how do you transfung the fungus as well as the small plant?
Yeah, good questions, thank you. So the benefits to the fungus. So we know in adult plants, there's been some good studies that show that the adult orchids pass carbon, so energy, to the fungi. So the fungi get the reward from that symbiosis, and the fungi pass carbon and nutrients to the plant as well. So it is, in adult plants, it's probably, well, at least of the ones that they've studied, is probably a truly sort of symbiotic relationship, or mutually beneficial. But up until the point where the orchids start photosynthesizing, they're not passing any carbon to the fungus. And we really have no idea what the fungi get out of it in that initial phase of germination. I mean, they don't get anything out of it that we can tell. Some people, particularly people who come from a plant mindset, think that the orchids must be tricking the fungi because orchids are obviously smarter than fungi, and that maybe they're sort of parasitizing them somehow, maybe. Or maybe the fungi get some benefit by being able to live inside the orchid tissue, like it's sort of a safe space where they can just hide out, and that might give them some competitive advantage in accessing those nutrients around the fungus plant. But yeah, in the germination phase, when you're talking about a tiny microscopic orchid seed, that probably seems unlikely. But so yes, they do get energy from the adult plants, but in seedlings, we're not sure what they get. That's kind of the limit of our understanding. And then secondly was, when we're reintroducing these orchids, we don't actually specifically inoculate the soil with the fungi, because we're growing the orchids with the fungi in those petri dishes. The orchids have the fungi inside their root tissue anyway. So when we plant them back out into the wild, we are planting the fungus as well as the orchid, because they're together. we did a review a few years old now, showing that what are the factors that influence the success of these orchid reintroductions or versus their failure. And one of the things is that growing orchids with their fungi is a significant factor in the success or failure of the reintroduction. If you don't grow the orchids with their fungi, which you can do using special chemical mixtures, which is how a lot of orchids are grown commercially for the flower industry and stuff. But when you plant those orchids out into the bush, they tend not to do very well. So the growing orchids symbiotically with their mycorrhizal fungus is actually really important to the success of them when you plant them back out into the wild.
Yeah.
Thank you for your talk. It's really good. I not missed it. Do those fungi exist in the wild without the orchids?
Oh yes, good question. These are all things I should have put in my talk, but yeah, they do. Well, at least many do. You can take those tea bags and plant them not near the base of wild orchids, but just out in random spots in the bush where there's no wild orchids, and sometimes at much lower rate, but you do sometimes get germination. And we also saw from that slide with that map, colourful map of Australia, that the CSIRO, through this big soil project where they've tested the DNA of soils all around the country, have found these orchid mycorrhizal fungi just in bits of soil that they've collected, not in orchids. So the fungi, most of these fungi probably don't need the orchids, we don't think. They're just hanging out in the soil doing their thing. Or on trees, most orchids don't grow in the ground. Most of the 28,500 species of orchids grow as epiphytes on trees in rainforest, and their seed lands on the bark of a tree in the rainforest, and the fungi that live on that bark then germinate the seed. So, yeah, the fungi are there kind of anyway. They may get some benefit from associating with the orchids, but yeah, they don't need them to survive.
There's a question here.
For those underground orchids, well, they live underground and they're white, which I suppose means they don't photosynthesize. So does that mean that the fungi are providing the orchid with their carbon as well as the nutrients? And if so, do you know how that happens?
Yes, so they associate with, it must be well, it must be, maybe it's not. They don't provide any, they don't photosynthesize, so they don't provide any nutrients to their fungi. And so we assume that that's a parasitic relationship or something of the sort. That may not be strictly true, because maybe the fungi get something else, like a safe space to live in the root tissue of the orchid where they can, and that's helpful to them. But that's often they associate with different species of fungi to other orchids. So it's a bit, I think it depends on which orchids you're, around the world that you're looking at, but possibly parasitic more than a truly symbiotic association.
Just actually, right, Sally.
Yeah. Hi, Mark. I'm interested in the fact that the fungi that help to germinate leek orchids require more nutrients than the fungi that help to germinate spider orchids and diurus and all those other things. And I guess the leek orchids that live around here live in quite fertile environments compared to, say the orchids of the box ironbark or coastal heaths or whatever. And I just wonder whether there's meaning in that.
Yeah.
And I also kind of wonder whether there's a role for fire in all of this, because obviously fire would have been traditionally used where all these orchids grow on a very regular basis, because they're basically in grasslands or woodlands.
Yep.
Are you kind of able to say
Oh, yeah, well, yeah. So I think you're probably right. And a lot of leek orchids are very fire responsive, so you're often only, some leek orchid species, they only ever flower after fire. Others will flower better after fire or whatever. And that is probably related to the pulse of nutrients that is released into the soil after a fire, and the fungal activity is probably is higher, the fungi are more active, and everything's just, all systems go, and they pass a lot of that to the orchids, and the orchids flower. And yeah, it's interesting, we've now been germinating quite a lot of different leek orchid seeds, and species of leek orchids from sandy soils, this is possibly a generalisation, but some species of leek orchids from sandy soils don't like that higher nutrient medium to germinate on. They actually only germinate on a different lower nutrient medium, so there probably is some signal there where, and it makes sense that the fungi from more fertile soils out on the plains around here are probably used to having a high level of nutrients in their environment than fungi from some sandy soil somewhere. But a lot of these fungi then eat leaf litter, don't they? So maybe that doesn't make any sense. Yeah, probably.
So, just when you were saying that the underground, I've lost the word, the underground flower plant doesn't feed the fungus, can you make a big leap from that to say that fungus can only survive on products of photosynthesis?
No, because these fungi are found in soils free living all around the place. Yeah, they chill out just fine. And yeah, yep. So it is a bit unsure why they associate with a species of orchid that can't provide them with any food.
Maybe they think they're really beautiful.
Yeah. Yeah.
Or it's a habit, maybe it's just a habit.
Yeah, yep.
And then they've lost the leaves.
There's one question.
Oh, yes. No, I was wondering if you want to be involved in going out and planting these new little orchids. Do you go down to Cranbourne and get trained and then get sent to particular places? Or do you have to be part of a team with the experts telling you exactly where to go and be carefully supervised?
Yeah, I wish it was the former. That sounds like a much better way of running things. Organised and, yeah, no. It's most of the volunteers who plant our orchids are from the Australasian Native Orchid Society. So get in touch with them. There's people, they have different groups. There's people who love to grow epiphytes in tropical orchids in their backyards. I tend not to speak very much to those people. But the other part of that organisation is people who are involved in conservation activities. And they do lots of cool stuff, surveying in particular, and helping us to plant orchids. And and they do trips all around the place, often to, up here. So if you're involved with I think it's like $20 a year is the membership or something, and they've got lots of activities on, and you can just sign up to whatever takes your fancy. But they're a really great group of people. Yeah.
Thank you.
Oh, hang on. 6 at once.
Orchids as a group are a very recently evolved group, and one assumes that to some extent the fungi have evolved along with them, especially the ones who've got particular relationships. But fungi don't present very good specimens for fossils. So is anything known about in the fossil record about the relationships of orchids and fungi?
Oh, that's a good question. I feel like that's a question for a real mycologist. I'm not sure, to be honest. Yeah, we do see those patterns though between, you know, the spider orchids associate with this genus of fungi and the leek orchids associate with this other genus of fungi, and there must be some, yeah, evolutionary cause for that. And I think I've read papers about that before, that, you know, there are patterns there that suggests something like that. Some sort of coevolution might be pushing it, but yeah. But yeah, that's not my area of expertise, sorry.
A lunchtime conversation.
Yeah.
A couple of others.
Do you use like iNaturalist records to be able to find, like, as us maybe not getting involved with these groups, can we use that as a citizen science tool?
Yeah, sure. iNaturalist is great and very helpful. The tricky thing with orchids, though, and with other groups of organisms that too many people have spent too much time studying them, is that they can be quite difficult to identify. And often you really need to like look at under a microscope at the particular parts of the flower, and it can be hard to tell from a photo. Sometimes you can tell. Sometimes if there's a white leek orchid from the plains around Euroa, you know it's going to be this particular species because that's the only white leek orchid from this area. So yeah, yes and no. Sometimes it's not quite scientific enough, but often it is. So yeah, that's a really cool thing.
If we were using like macro lenses and trying to get these photos,
yeah. Particularly if you know, well, if you. The good thing about orchids and other plants is that there's keys so the Botanic Gardens has this Vic Flora page, which has got all the species of plants in Victoria, and identification keys, and you can actually go through and say, oh, this one has whatever part of its flower is this long, and therefore it's this species. So if you know the part of the diagnostic feature of that species and can photograph that, then that's really helpful. Yeah. But yeah.
There was another question.
Just, you talk about a lot of cryptic orchids and they're growing, and I'm aware. Could you just want to speak to hyacinth orchids and their sort of difficulty around cultivation? Because I know you can get them in petri dishes and grow them not out.
Yeah. So hyacinth orchids are those pink ones that flower in summer. I had a photo of it at the start. Not very good. And they are one of the few groups of orchids in Australia that don't associate with these big fungi genera, the ones I've put up before. I think the hyacinth orchids actually do associate with ectomycorrhizal fungi, maybe russulas or something, I'm not sure. So the fungi that are in those orchids need the living tree to survive. And we've had way too many pots of hyacinth orchids brought into us, and there's a lovely hyacinth orchid there, but you know it's only a few days until it dies, because its fungus actually requires that living eucalypt, tree that it was growing next to. And so yeah, they're very difficult to grow. We can get the seed to germinate in a special asymbiotic mixture if we match the right chemicals and hormones and things, but they don't. Without their fungi, they can't do anything. So that's yes, some fruitful ground for a future PhD or someone to work out how to grow those, because there are a couple of quite rare species of hyacinth orchids. Yeah.
We've got time for one more question. Can I ask one?
Sure.
At the risk of asking the botanist a mycological question. So you had a slide up there, Mark, where you had the relationship between particular species of orchid and a number of different fungi, and all of them associated with that fungus. And they're identified using basically sort of genotypal. So, we're all similar genotypes in this room, but we all have different behaviours. And more identity of cryptic organisms is going using genetic techniques. So with fungi, do fungi have particular, I'm now sort of addressing this more broadly, are there behavioural differences
Yeah.
between the way fungi of the same genotype might respond to different things, and
Yep.
so is a genetic ID necessarily
It's limited, isn't it? Because that's how we identify these orchid mycorrhizal fungi, because they don't produce visible fruiting bodies. The only way
what are you going to do?
we can identify them is through their DNA. And that's helpful to a certain degree, but yeah, you're right. When we look at different individuals that all have the same DNA sequence for the particular region of the DNA that we are looking at, we do see differences. You know, like some of them use that higher nutrient medium and some of them do better on the lower nutrient medium. We also see morphological differences, like there were 3 isolates in that, 3 individual fungi that in that slide I was talking about. And some of them, one of those fungi would grow all the way up into the air, like all through the petri dish, up all the leaves of the orchid seedlings as they were germinating, whereas the other fungi would stay only on the surface of the agar. So obviously, you know, we require a lot more. We're just at the very start of that journey into using DNA to identify fungi. Like we've got it to the point where we can easily identify what are different species of fungi. But you're right, there's a lot more. Fungi, even individuals of the same species, can be very different in their behaviour and how they react in the petri dish in the lab, and probably how they react in the wild. So, yeah.
And what is a species of fungus?
Yes.
Maybe somebody else is going to address.
Well, we refer to them as operational taxonomic units, which.
Right then.
Yeah.
Can we thank Marc for a wonderful presentation?